Goji Guy, YouTuber, dinosaur hugger, kaiju lover, father of Hank, and now father of Vex. Today I challenge Goji Guy saltwater beluga glass orca shark Saurus with a giant shark. Sharks are apex predator of the ocean, dominated the food webs for over 400 million years. Their hydrodynamic body shape, cartilage skeleton, and razor sharp teeth are the gold standard in their evolution. Some shark species like great white shark, hammerhead shark, bamboo shark are some predators of the ocean. Sharks continuously replace teeth, heal wounds quickly, and resist cancer in a way. Their unique immune system combats pathogens in open ocean environments. Part 1. Megalodon. Our foundation is Otodus megalodon, king of ancient seas. Fossil teeth first appeared about 23 million years ago in the Miocene, and megalodon ruled oceans until roughly 2.6 million years ago. Its lineage gives us the genetic blueprints for massive growth, cartilage durability, and predatory edge. When we talk height, we mean the vertical thickness of the body, from belly to dorsal fin. Estimates place Megalodon at around 3 to 4 meters tall. That towering profile helps our hybrid cruise just below the surface, shadowing prey from above. Fossil reconstructions peg Megalodon's maximum length between 15 and 18 meters. That's nearly 3 school buses end to end. In our design, those variants are what push modern shark skeletons toward that colossal scale. At full size, Megalodon tipped the scales at an estimated 50 to 65 metric tons. From its genome sets our hybrid's mass potential or massive potential, muscle bulk, and bone density to match. Scientists estimate Megalodon delivered between 108,000 and 182,000 newtons of bite force, enough to crush a small car. Jaw muscle myosin variants and reinforced ligament to recreate that crushing power. Megalodon had roughly 276 teeth, arranged in 5 rows per jaw. Each tooth was replaceable. When one fell out, another rolled forward. We mimic that tooth replacement system by editing key developmental genes for continuous dentition. Individual Megalodon teeth could exceed 18 centimeters in height, bigger than your hand. By integrating genes for enamel hardness and dentin growth, our hybrid will sport similarly enormous self-sharpening razor. Finally, Megalodon's jaw gape could exceed 2.7 meters, wide enough to engulf large whales. We target the wide-angle hinge, giving our hybrid maximum bite aperture. Part 2. The Arapaima. In the Amazon rivers, these giants patrol oxbow lakes, flooded forests, and backwater channels where oxygen levels plummet during dry seasons. In these oxygen-poor, tannin-stained waters, Arapaima surface every 10-15 minutes to gulp air. Their range spans 6 countries: Brazil, Peru, Guyana, Suriname, French Guiana, and Venezuela. Arapaima scales aren't your typical fish scales. They're composite structures, a tough outer layer of ganoid and enamel-like mineral, and a thick inner layer of elasmodene, a collagen-rich protein matrix. Part 3. The scaly-foot gastropod. This little snail lives where few creatures dare. 2,400 meters down at hydrothermal vents along the Indian Ocean ridges. Temperatures swing from near freezing to boiling in meters, and metal-rich fluids gush 24/7. It's here the scaly-foot gastropod evolved its miraculous armor. Unlike any fish or reptile, this snail's scales are true armor plates, hard as mild steel. But built from biological material, impact tests show they absorb and dissipate twice the energy of a rhinoceros beetle's shell, making it the toughest natural armor we know. The secret sauce is a unique bacterial partner. Gamma proteobacteria living in the snail's outer layer feed on dissolved iron and sulfur, precipitating iron sulfides right into the shell. Without these microbes, the snail couldn't build its metallized armor. Each armor plate has 3 precise layers. First, the outer shell of iron sulfide minerals, providing metal-like hardness. Second is middle organic periostracum, a leathery protein film that glues and cushions. And third, the inner aragonite layer, a crystalline calcium carbonate that resists cracking. This sandwich structure balances rigidity, toughness, and flexibility. Now imagine layering Arapaima's thick bony plates with Scalyfoot's iron sulfide lamellae twice over. you'd end up with living chainmail backed by a steel plate. Our giant hybrid shark would shrug off predator attacks, falling debris, even high-velocity projectile impacts. In short, a mobile fortress of fish and snail biology. Art 4. Manta rays. These rays aren't just majestic, they're brainiacs. Scientists rank them as the most cognitively advanced fish, even rivaling some mammals in problem-solving and learning speed. Today, we're stealing their secret sauce to make our giant hybrid shark not just powerful, but brilliant. Most fish have tiny brains, just 0.1% of body mass. Mantas push that up to nearly 0.3%, matching dolphin. For a 5-meter wingspan ray, that's a huge headspace for memory, spatial mapping, and creativity. Mantas sport an oversized cerebellum for fine-tuned swimming and a highly folded telencephalon for advanced processing. In mirror mark tests, only a handful of non-mammals pass, and manta rays ace it. They recognize themselves, indicating self-awareness. That's a trait we want in our shark so it can adapt, learn from failures, and even exhibit curiosity. Manta rays form long-term social bonds, coordinate group hunts, and remember safe cleaning sites. By adding their social brain gene modules, our hybrid shark could strategize with pod members or even learn cooperative hunting tactics. Brute strength alone makes a predator, but intelligence turns it into a strategist. A mega shark with manta-grade brains won't just charge blindly. It'll plan ambushes, adapt to new environments, and avoid harmful conflicts with humans or its own kind. Smarter means safer, at least inside our containment. And far more fascinating to study. Part 5. The black-spotted tuskfish. Meet Chirodon sherwinlinii, the black-spotted tuskfish, number 2 in our intelligence lineup. Their brains may be smaller than mantas is, but they rival primates in behavioral flexibility. They'll carry tool shells, rocks, up to 50 body lengths before using them. That spoke of mental planning, choosing a tool, transporting it, and remembering where to strike for maximum effect. When their path is blocked, tuskfish try multiple angles until they succeed. They don't stick to one strategy, they adapt on the fly, showing true cognitive flexibility. They remember precise locations of food caches and shelters even after days. That long-term spatial memory is crucial for our giant hybrid when mapping vast ocean territories. In lab studies, tuskfish learn a new task in just a few trials, faster than most teleosts. They even invent novel methods to bypass obstacles, a hallmark of true innovation. A purely aggressive shark is predictable and dangerous. By adding tuskfish foresight and flexibility, our hybrid becomes an adaptive hunterable to navigate complex reef structures, avoid booby traps, and even learn from other sharks. Smarter means safer, both for the shark in containment and for the ecosystems we study. Part 6. Atlantic salmon. After manta rays and tuskfish, Atlantic salmon claim the next tier of fish smarts. Their brains may look small, but these silver-sided navigators show us what real-world problem-solving and adaptability look like in action. Each year, salmon hatch in freshwater streams, migrate thousands of kilometers to the ocean, then return to the exact gravel bed they were born in. They use geomagnetic maps, olfactory landmarks, and even the sun's position, an unrivaled internal GPS. When presented with new dams or fish ladders, salmon quickly learn the correct channels. They remember gate positions, water flow patterns, and shift tactics on the fly. Real-time spatial reasoning in swirling currents. Salmon transition seamlessly from fresh to salt water and back again. Their brains rewire to handle different salinity, predators, and food sources. That kind of neuroplasticity means rapid learning in new environments. Salmon transition seamlessly from fresh to salt water and back again. Their brains rewire to handle different salinity, predators, and food sources. That kind of neuroplasticity means rapid learning in new environments. Salmon migrates in synchronized schools, choosing leaders who break the hardest currents. Younger fish follow veteran paths, showing coordinated teamwork and social learning. They integrate pressure waves, chemical gradients, and light cues with extraordinary precision. That multimodal sensing lets them avoid obstacles, locate hidden riffles, and detect prey or mates at a distance. A brute force predator can smash through prey, but an adaptive predator thinks its way through challenges. We'll gift our hybrid shark the memory, flexibility, and leadership to navigate novel reefs, coordinate hunts, and even solve containment puzzles. In short, smarter means more control, more efficient, and far more fascinating to study. Part 7. Pacific Sleeper Shark. Deep below 1,000 meters, where sunlight vanishes, the Pacific sleeper shark hunts. It boasts the most developed night vision in all of marine life, better even than some nocturnal mammals. Their retina is packed with rod cells, photoreceptors specialized for low light. Pacific sleepers have rod densities up to 10 times higher than surface sharks. Sleeper sharks have eyes nearly twice the diameter of similar-sized species. Their pupils dilate to a massive aperture, maximizing that rod-rich retina's field of view. Behind the retina sits a mirror called the tapetum lucidum. It bounces stray light back through the rods for a second chance at detection. Pacific sleepers have one of the thickest tapetum layers known, amplifying faint glows from sinking carcasses or bioluminescent prey. The sleeper's spherical high-refractive lens corrects for light distortion at extreme depth. It focuses dim beams sharply onto its rod-packed retina, avoiding blurs that defeat other deep-sea eyes. Our hybrid swims from sunlit shallows to midnight trenches. Upgraded, the sleeper shark eyes ensure it never loses sight of prey or obstacles, whether stalking squid at 2,000 meters or cruising twilight reefs. Part 8. The Nile Tilapia, a hardy cichlid native to African lakes and rivers. You've probably tasted them on your plate, but beneath those scales lurks one of the most advanced fish eyes on the planet. Over a split screen of a tilapia eye and a human eye, tilapia evolved in clear, shallow waters full of plants and prey. To distinguish rippling shadows, ripe fruits, and colorful algae patches, they developed trichromatic vision just like us. They express 3 cone opsins tuned to long, red, medium green, and short, blue wavelengths. Together, these pigments let tilapia see a broad palette of hues under sunlight-filtered water. The tilapia's crystalline lens transmits 350 to 700 nm without major absorption. That means ultraviolet through red light all reach the retina with minimal loss, critical for full-spectrum color detection. After phototransduction, signals feed into opponent channels, red versus green and blue versus yellow. This neural subtraction enhances contrast between similar hues, enabling precise color judgments. In lab trials, tilapia reliably learned to peck red, green or blue targets for food, even under changing light levels. That behavioral evidence seals the deal. They're true trichromats. Most sharks see in muted blues and greens only. By gifting our hybrid full-spectrum color vision, it can detect cryptic prey, spot warning displays on reef fish, and navigate complex coral landscapes with human-level hue discrimination. Part 9. Blue damselfish, tiny reef guardians with vision sharper than you'd expect. These little dynamos navigate sun-drenched shallows where color matters for everything from mating signals to predator alerts. Tiny reef guardians with vision sharper than you'd expect. These little dynamos navigate sun-drenched shallows where color matters for everything, from mating signals to predator alerts. Blue damselfish have 3 specialized cone cells: short wavelength sensitive, SWS1, medium, HH2, and longae. Together, they allow this fish to split sunlight into a kaleidoscope of colors, perfect for high-resolution reef life. Part 9. Blue damselfish, tiny reef guardians with vision sharper than you'd expect. These little dynamos navigate sun-drenched shallows where color matters for everything, from mating signals to predator alerts. Blue damselfish have 3 specialized cone cells: short wavelength sensitive, SWS1, medium, ARH2, and long data to allow this fish to split sunlight into a kaleidoscope of colors, perfect for high-resolution reef life. Their eye lenses are ultra-clear, made for full-spectrum input. They pass both UV and visible light with minimal distortion, vital for picking out UV reflective patterns or subtle pigment changes in other reef species. From deep violet to fiery red, the damselfish eye is built to transmit a wide range of wavelengths. Their lens material avoids UV blacking proteins found in many fish species. Unlike humans, who have overlapping cone sensitivities, damselfish have tightly tuned opsin response curves. That means less spectral blurring and finer distinction between even similar hues like turquoise and blue-green. Like Nile tilapia, damselfish process color contrast through opponent pathways: red versus green, blue versus yellow. This heightens edge contrast and lets them zero in on patterns and motion, even in complex visual clutter. Behavioral trials show damselfish can consistently distinguish 3 primary hues, even when brightness is equal. That confirms true trichromatic capability, not just brightness-based guessing. Each species brings a unique color-processing edge. Tilapia offer human-like balance across the spectrum, but damselfish bring razor-sharp mid-spectrum tuning, ideal for detecting prey, warning coloration, and camouflaged predators. Together they give our hybrid shark precision color vision across reef, coastal, and open ocean environments. Part 10. Zebrafish. The zebrafish may only be a few centimeters long, but its eyes are a marvel of biological engineering. Native to the rivers and rice paddies of South Asia, these little swimmers have become the go-to model for vision science. And it's easy to see why. Like us, zebrafish are active during the day and rely heavily on vision for navigation, foraging, and social interaction. But unlike humans, they don't stop at red, green, and blue. They see ultraviolet light too. Zebrafish express 4 distinct cone types: UV sensitive, SWS1, blue layout, expands their perceptual palette beyond the human visual spectrum. Color processing begins before the brain even gets involved. Opponent cells in the retina subtract UV from blue or green from red, sharpening contrast and enhancing pattern recognition before higher level interpretation kicks in. Behavioral tests confirm what the physiology predicts. Zebrafish learn to associate UV markings with food or mates. They don't just sense UV, they use it intelligently to survive. Most prey species reflect UV in subtle patterns invisible to traditional shark eyes. By borrowing zebrafish's UV opsins and lens design, we allow our hybrid to decode hidden signatures, track stealthy organisms, and distinguish friend from foe under moonlight. Part 11. The American Shad. This is Alosa sapidissima, a migratory herring that travels from ocean to freshwater to spawn. They're famous for explosive schooling behavior and, as we'll see, an uncanny ability to eavesdrop on the world around them. Though we think of fish as nearly deaf, shad rank among the top in aquatic sound detection. Their inner ears pick up faint pressure waves and vibrations that most other fish completely miss. Shad have swim bladder bulli, air chambers that press directly against the ear bones. When sound waves hit the bladder, it vibrates like a drum, transmitting amplified signals straight to the inner ear. A labyrinth of water-filled cranial canals links the ear to pressure sensors along the flanks. This network funnels minute pressure changes from across the body into the auditory system, boosting sensitivity across multiple points. Inside, tiny hair cells cling to the otolith. When the skull vibrates, the dense otolith lags behind, bending the hairs and converting motion into neural signals. Shad pack more of these hair arrays per square millimeter than nearly any other fish. Shad hear from low bass rumbles to ultrasonic clicks, roughly 30 Hz to 15 CHs. That range covers everything from thunderous storms to snapping shrimp, making them acoustic generalists in both river rapids and open ocean. In tight schools, everyone's swim bladder vibration merges into a collective chorus, amplifying faint sounds and creating an underwater megaphone. Imagine a 12-meter predator that not only sees in darkness, but also hears approaching prey or vessels from hundreds of meters away. With Shad's bulle-coupled ears and canal network, our hybrid pinpoints movement in murky water, eavesdrops on distant schools, and even senses mechanical vibrations from human submersibles. Part 12. Great White Shark. Meet Carcharodon carcharias, the great white shark. At up to 6 meters long and 2 tons heavy, it's the ocean's ultimate hunter. With streamlined bodies, serrated teeth, and unmatched sensory systems, great whites have ruled nearshore food webs for millions of years. In this segment, we're harvesting their signature senses to upgrade our hybrid colossus. Great whites can detect blood concentrations as low as 1 part per 10 billion. Imagine a single drop in an Olympic-sized pool. Their nostrils feed 2 giant olfactory bulbs in the brain, each lined with hundreds of millions of receptor neurons. That extreme smell lets them track injured prey from kilometers away. Ampullae of Lorenzini are electroreceptors tuned to microvolt level voltage gradients. Great whites have over 7,000 pores packed across their snout and head. By sensing the bioelectric fields of muscle contractions in prey, or even the Earth's magnetic field, they navigate, hunt buried animals, and orient themselves across vast ocean distances. Running down each side is the lateral line canal system, pressure detectors built from fluid-filled networks and hair cell clusters called neuromasts. Great whites use this to feel minute water movements, a fin's ripple, a distant splash, or the wake of a school of fish. Combined with smell and electrosensing, the lateral line completes their trifecta of ocean mastery. Part 13, Mexican tetra. This is the Mexican blind cavefish, Astyanax mexicanus. Descended from surface-dwelling ancestors, these fish have spent millennia in dark limestone caverns beneath Mexico, evolving without eyes, yet thriving in total darkness. Despite losing their eyes, cavefish boast one of the most remarkable mechanosensory systems on Earth, their lateral line. Compared to surface fish, cavefish pack more neuromasts per millimeter, span longer head-to-tail canals, and detect flows down to micrometer scale disturbances. No other vertebrate senses water motion this precisely. Neuromasts are tiny hair cell sensors in fluid-filled canals. Cavefish neuromasts are not only larger, giving them greater sensitivity, but crammed in dense arrays, so overlapping fields cover every inch of their bodies. Studies reveal cavefish have higher baseline firing rates in lateral line neurons. That constant hum of electrical activity primes them to detect the slightest current change, like moving one grain of sand. Most fish dampen lateral line signals when they swim to avoid self-noise. Cavefish lack this efferent inhibition, so they hear every ripple they create, turning self-generated flows into sensory feedback for navigation. Combining dense neuromasts, high firing rates, and no self-inhibition, cavefish build real-time flow maps of their caves. They detect overhead ledges, tiny overhangs, and prey hiding behind pebbles, all without a single eye. Interestingly, other blind cave dwellers, from salamanders to crustaceans, evolved similar enhancements. That convergence tells us this sensory upgrade is a robust blueprint for navigating darkness. The three-spin stickleback may be small, just 6 centimeters long, but it punches above its weight in sensory adaptation. Found in both marine and freshwater environments around the northern hemisphere, this fish is a model of evolutionary ingenuity. Part 14, three-spined stickleback. Sticklebacks sense water flow with extraordinary precision. Their lateral line system rivals that of cavefish and blind salamanders, detecting pressure gradients down to micrometer scales. That makes them masters of navigation and predator avoidance. Unlike many fish with deep lateral line canals, sticklebacks trade canal depth for surface neuromasts. These exposed hair cell clusters sit right on the skin, supplying instant feedback on turbulence and directional flow. Independent stickleback lineages, one in lakes, one in coastal waters, both boosted neuromass density in parallel evolution events. It's nature's proof of concept. More sensors, better flow mapping, faster reaction. Scientists have identified quantitative trait loci controlling neuromass number, spacing, and regeneration. By isolating these genetic modules, we can selectively amplify the stickleback's best lateral line traits in our hybrid shark. Remarkably, sticklebacks, cavefish, and blind salamanders independently evolved similar lateral line enhancements. That convergence tells us this sensory upgrade is a robust blueprint for any creature navigating dark or turbulent waters. Some sticklebacks develop more neuromasts on one flank, creating sensory asymmetry. That leads to a handedness in turning behavior, another layer of adaptability when hunting or evading predators. In turbid or dark water, sticklebacks dial down vision and dial up mechanosensation. They sacrifice color cues for flow data. A trade-off we'll incorporate so our hybrid shark adapts seamlessly from clear shallows to murky depths. Great whites give raw range, cavefish deliver constant high-fidelity signals, and sticklebacks add surface sensitivity and adaptive asymmetry. Layering all three creates a lateral line super network, one that senses prey, obstacles, and even subtle current shifts from hundreds of meters away. Part 15, Black Ghost Knifefish. This is the black ghost knifefish, Aptonotus albifrons, a South American freshwater dweller with an invisible superpower. Today we're unlocking how it senses the world with electricity and why those genes belong in our giant hybrid shark black ghost knifefish boast one of the most sophisticated electrosensory systems known. They emit continuous electric signals and sense the tiniest disruptions, mapping prey, obstacles, and even fellow fish in complete darkness. Unlike electric eels that fire pulses, knifefish emit a continuous sine wave discharge around 800 to 1200 Hz. That constant carrier signal makes it easier to detect frequency shifts caused by nearby objects and living creatures. Tuberous electroreceptors, called normiromasts, are packed densely along the head and flank. Each receptor measures minute voltage changes down to microvolts, letting the fish feel the electric signature of its surrounding. Research pinpoints distinct quantitative trait loci that control EOD frequency, receptor density, and receptor placement. These modular genes are exactly what we splice into our hybrid shark to recreate knifefish-grade electrolocation. In lab tests, knifefish neurons fire reliably at voltage fluctuations as low as 0.01 vertebrates or slow-moving prey with unmatched precision. The electrosensory lobe in their hindbrain is massively expanded to process all that incoming data. More cortex devoted to electric vision means faster object discrimination and spatial mapping. Lacking scales reduces electrical noise, and the undulating ribbon fin propels them silently. No tail slaps means minimal self-generated disturbances, so the knifefish hears only external signals. They sacrifice vision in crepuscular or turbid waters to amplify electric sensing. A perfect trade-off. For our giant hybrid shark, adding knifefish genes means a secondary sixth sense that complements vision, smell, and pressure detection. By grafting knifefish EOD machinery and receptor arrays into our shark genome, Colossus will sense hidden prey and obstacles even in pitch-black depths. Part 16, Brown Ghost Knifefish. This is Apteronotus leptorhynchus, the brown ghost knifefish. It prowls South American streams and flooded forests at night, navigating absolute darkness with an electrical sixth sense. The brown ghost's electrosensory network rivals even the black ghost knifefish. It decodes microvolt distortions in its self-generated field to detect prey, obstacles, and fellow fish, without using its eyes at all. Unlike pulse meter electric fish, brown ghosts emit a steady high-frequency sine wave, up to 1200 Hz. That continuous carrier lets them monitor rapid changes in their environment, picking up moving targets or tiny eddies in real time. Brown ghosts also talk electrically. They modulate frequency and amplitude to signal identity, mood, or mating readiness. That rich electrocommunication repertoire hints at sophisticated neural control we can adapt for social or cooperative behavior in Colossus. These tuberous electroreceptors are packed densely along the head and sides up to 90 per square millimeter. They detect voltage changes down to a few nanovolts, giving pinpoint spatial resolution within a few centimeters. The brown ghost's brain devotes a massive chunk to the electrosensory lateral line lobe. This hypertrophied center rapidly integrates receptor input, filters self-noise, and constructs a dynamic electrical map of the surroundings. With no scales damping the field and organs running along its entire length, the brown ghost achieves full circumference coverage. It senses objects dorsal, ventral, or behind N, all-round radar that never lets anything slip by. Part 17, Hammerhead Shark. Hammerheads, Sphyrna, Speppel roam tropical and subtropical seas, cruising in giant schools by day and solitary at night. They're instantly recognizable by that wide cephalofoil, which isn't just a hammer-shaped head, it's a sensory superstructure. On each edge of the cephalofoil, tens of thousands of electrosensory pores detect minute voltage fluctuations caused by muscle contractions in other animals. By sweeping its head side to side, a hammerhead crafts a detailed electrical map of prey buried in sand or hiding behind coral. Interspersed with the electrosensors are ampullae of Lorenzini, specialized jelly-filled canals that sense temperature, salinity, and magnetic fields down to microvolt levels. Hammerheads pack over 20,000 pores across their broad snout, giving them extraordinary range and directional precision. In our hybrid's genome we've already borrowed great white's basic electrosensitivity and Lorenzini channels, black ghost knifefish's continuous EOD pulse and receptor arrays, brown ghost knifefish's high-frequency EOD and hypertrophied sensory lobe, and now Hammerhead's cephalofoil distributed sensors and ultra-dense Lorenzini pore. By layering these 4 electrosensory modules, our giant hybrid shark will detect prey buried beneath meters of sand, eavesdrop on the electrical chatter of schools, and even navigate using Earth's magnetic field, all in real time. It's the ultimate underwater radar. Part 18, Electric Eel. Meet Electrophorus electricus, the electric eel. Despite its name, it's a knife fish, not a true eel, and it's the electricity champion of the animal kingdom. Today, we're stealing its shock tactics for our giant hybrid shark. Electric eels pack up to 5,000 electrocytes in series each cell, can produce around 0.15 volts. When fired in unison, they unleash up to 860 volts in a split second, enough to stun prey or deter a chimera. Eels have 3 specialized electric organs. The Sachs organ delivers weak pulses for electrolocation. Hunter's organ produces moderate discharges for navigation and communication. The main organ fires the big 600-860 volt shocks for attack and defense. Each organ is a gene-regulated stack of electrocytes perfectly tuned for different tasks. Imagine a 12-meter apex predator that not only sees, smells, hears and senses electric fields, but can also deliver a massive jolt on demand. A built-in stun gun to immobilize prey in deep darkness. Defense against large threats from submarines to rival hybrids. A secure way to train and guide the shark in containment. Small shocks as behavioral cues. By grafting eel electric organs into a shark body, we give our hybrid an extra weapon and a Part 20, Introduction. Scientific name, Tulcanus scalatum, Tonitrua morsus, Otter's common dictionary name, volcano-scaled thunderbite shark. Nickname, Colossus. Female, counterpart, gigantic. Height, 12.5 feet. Length, 33.52, 58.7 feet. Weight, 60 metric tons. Physical characteristics. The similar size and body of the megalodon, dermal plates of a rapaima and scaly foot gastropod layered for steel-like hardness. Oversized eyes with rhodopsin from Pacific Sleeper Shark for pitch-black vision. Tetrachromatic cone array plus UV-sensitive opsins for full-spectrum sight. Ampullae of Lorenzini and 4 Specky's electrosensory network for electric and magnetic mapping. 3 electric organs derived from electric eel for 800-volt discharges. Behavioral traits. Cooperative. Pack hunting strategies guided by manta ray and tuskfish cognition. Spatial memory rivaling salmon for migratory navigation in vast enclosures. Tool-like use of electric pulses to stun prey or signal conspecifics. High sensory plasticity shifts between vision, hearing and mechanoreception depending on environment. Speed of swimming. Cruise 20 miles per hour, burst 30 miles per hour. Bite force 150,000 newtons, enough to crush the hull of a small submersible. IQ. Equivalent to a bottlenose dolphin or great ape, roughly 120-130. We've stacked Arapaima's bony elasmodine backing with the scaly foot gastropod's iron sulfide faceplates. The result is living armor that outperforms many engineered materials. Let's break down the numbers. At 4.5 to 5.5 GPa, the surface hardness rivals mild steel, next 0.4 GPa, and even some tool steels. That means threats like sand abrasion or rasping teeth barely scratch the shell. With compressive strengths approaching 10 MPa, Colossus's armor can bear pressures equivalent to a compact car balanced on a fingernail without yielding. A tensile strength of 200-300 MPa means the armor resists ripping apart under tension. For comparison, high-grade aluminum alloys sit around 300 MPa, so this is no flimsy scale. Kick values above 2.5 MPa to the power of 0.5 tell us cracks won't run. The tough elasmodine layer arrests crack growth, preventing brittle failure even under repeated stress. Thanks to the composite design, impact toughness doubles or triples that of raw iron sulfide. High-speed impacts like collisions with debris or predator bites are absorbed rather than shattering the plate. With hardness, strength, and toughness metrics exceeding many metals, penetration resistance can stop high-velocity projectiles up to rifle round energy ~2 kJ, bite/rake resilience, scales won't spool or crack under repeated puncture, survives predator attacks. Wear resistance, surface hardness rivals tool steel, resisting abrasion from sand, rock, ice. Self-healing potential. The Arapaima collagen matrix regenerates microdamage over weeks. Colossus patrols the deep like a living battleship. This is Colossus's Vision Suite, blending sleeper shark night sight with tetrachromatic combs from tilapia, damselfish, and zebrafish. Today, we'll break down just how far and how finely it sees, from pitch black to dazzling daylight. Detection threshold down to ~10.8 lux, starlight level. Rod density approaching 150 zero rods/mm/m in Pacific sleepers. ~100/UTVAN/M in Pacific sleepers. ~100/ human dark vision. 4 cone types cover 350, 700 nm UV, blue, green, red, spectral discrimination, tilde 10 mm precision, twice the fineness of human vision, dynamic range greater than 14 stops, purpose glare to shadow transitions without washing out. Photopic acuity ~20 cycles/degree hawk, level detail, scotopic acuity ~5 cycles/degree, enough to silhouette moving prey under moonlight. Results feature down to ~5 cm at 50 m distance. Flicker fusion threshold ~80 Hz, humans ~60 Hz. Tracks rapid fin beats, bioluminescent pulses, and explosive escapes in real time. Minimizes motion blur in swirling currents. UV channel 350, 400 nm. Reveals hidden biofluorescence and cryptic prey signatures. Polarization sensitivity enhances contrast of silvery scales and polarized glare patterns. Combined cues give Colossus an extra layer of light information invisible to other predators. With night vision that outperforms mammals and color vision richer than ours, Colossus dominates every light condition. This is Colossus's hearing system, supercharged by American shad genes and scaled up to megalodon dimensions. Today, we'll break down just how far and how finely Colossus listens. When we scale the American shad from about 0.5 m to Colossus's 15 m length, we get length 30, surface area equals 30 dxl equals 900, volume equals 30 dxl. That transforms the shad's swim bladder ear into a super ear. Resonance frequency scales inversely with radius. A normal shad hears down to ~30 Hz. Colossus's 30x larger bladder drops its low end limit to ~1 Hz, unlocking infrasound detection of storms, whale migrations, and submarine wakes. Surface area scales XX900, volume X270. Combined, that yields approximately a +75 dB boost in pressure sensitivity if a shad's threshold is 70 dB Ray 1 to 1 pp. Colossus can drop to 0.5 dB Ray 1 to 1 pp at resonance. The enlarged bladder not only lowers the bass cutoff but also extends the high end. Colossus's hearing spans roughly 1 Hz to 8 kmh, covering everything from deep rumbles to mid-frequency biological calls. Sound spreads with 20 log-R losses. A 75 dB gain means ~60 to detection distance. If a shad tracks a motor at 1K, Colossus hears it at ~60K. Low-frequency whale calls may carry over 100K. With infrasound to ultrasonic coverage, sub-nanopascal sensitivity, and tens of kilometers range, Colossus turns the ocean into a symphony of distant cues. In short, by blowing the shad's ears up to megalodon proportions, you create a super ear that hears like a submarine hydrophonable to pick up the faintest pressure waves from tens of kilometers away and down to frequencies below 1 Hz. But today, we're diving into something more primal: scent. Sharks rely on smell to track prey across kilometers, and Colossus takes that to a whole new level. Great whites have ~0.2 m dots of olfactory surface. Scale that to a 15-meter shark using Megalodon math, you get over 1.2 ME6 times the detection space housing roughly 180 million olfactory neurons. That's more than double a bloodhound's nose. Underwater, most sharks can smell 1 part of blood in 10 billion parts seawater. But with Colossus's expanded lamellae and enhanced sensory pathways, the detection limit drops to 1 in 100 billion. That's 1 droplet in 5 billion liters, a cube of ocean 170 meters on a side. Thanks to its size, Colossus inhales tilde-10 per second per sin, compared to every 3-5 seconds in smaller sharks. That means it refreshes odor samples every second compared to every 3-5 seconds in smaller sharks. It doesn't just smell better, it smells faster. Its nose isn't just tuned to blood. It detects amino acids, bile acids, and even lipid breakdown products from stressed fish or decaying matter. With expanded receptor subtypes, Colossus senses a broad chemical spectrum of life and death at sea. In open ocean, odor plumes dilute rapidly. Most predators lose the trail after 1-2 kilometers. But with its ultra-low threshold and massive sampling rate, Colossus follows scent trails up to 10 kilometers, threading scent ribbons in moving currents like a needle through fabric. Track bleeding prey across entire continental shelf edges. Detect chemical leaks or pollution sources. Find whale carcasses from miles away. Follow schools by trace metabolites they leave behind. Smell is the oldest sense in the animal kingdom. And now, Colossus wears the crown. By scaling up lamellar surface and neural circuitry, the hybrid's nose becomes tilde 6x more sensitive, cuts detection thresholds by an order of magnitude, and extends its chemical tracking range 5-fold. Colossus's lateral line isn't just big. It's built from 3 evolutionary supermodels: great white canals, Mexican tetradense neuromasts, and stickleback surface sensors. Together, they turn its body into a living hydrodynamic radar. We've integrated deep canals and pores from great whites for long-range pressure waves, enlarged densely packed canals and constant flow neuromasts from cavefish, expanded superficial neuromast fields from sticklebacks for high-resolution surface. By merging cavefish baseline firing with stickleback offset tuning, Colossus senses water movements down to tilde 0.005 mm/s. That's 20x finer than a normal shark, able to pick up the slightest fin flick from meters away. This superlateral line covers 0.01 Hz up to 500 Hz. It hears slow, infrasound-like currents and fast turbulence mapping, everything from tidal shifts to tiny prey fluttering. With neuromasts every centimeter and canal pore every few meters, Colossus localizes flow sources within tilde 1 dg accuracy and tilde 1 cm spatial resolution. Imagine pinpointing a shrimp hiding behind coral. Long-range canals detect large wakes and moving schools at 100 to 200 m. Superficial mats map obstacles and prey within 5 to m in high detail, giving both macro and micro-oceanic vision. Cavefish genes remove efferent inhibition, so Colossus never blinds itself in motion. Stickleback-style lateralization lets it favor one flank for targeted hunting, and advanced neural filtering rejects tailbeat noise. In clear water, it dials vision up and mech anosensation down. In turbid depths, it suppresses vision and cranks lateral line gain to maximum. This sensory plasticity makes Colossus equally lethal in every ocean zone. By fusing these 3 lineages, Colossus's lateral line becomes a living hydrodynamic radar, sensing the faintest current drifts centimeters away, mapping obstacles and prey tens of meters out, and tracking large wakes hundreds of meters distant with millimeter-scale precision. Colossus's electrosensory suite combining 4 apex designs. Today we quantify just how finely it reads electric and magnetic cues underwater. Detection threshold ampullae detect fields down to tilde 0.5 nanovolt/centimeter compared to tilde 5 nanovolt/centimeter in wild sharks. Knifefish electrolocation picks up microvolt level disruptions around its self-generated field. Frequency bandwidth DC to 20 Hz for static bioelectric and geomagnetic sensing. 200 Hz to 1000, 200 Hz for active electrolocation and knife-fish-style EOD signals. Spatial and directional resolution localizes targets to within ~1 cm at 1 m distance. Angular accuracy ~0.5.1 cell, letting Colossus map buried prey or hidden obstacles with precision. Signal processing expanded electrosensory lobes integrate greater than 4x more channels than any single species. Advanced neural filtering cancels self-noise, tail beats, and isolates external fields. The base pause of the great white is 7.0, which scaled approximately ~43,800. The base pause of the hammerhead is 20.0, which scaled approximately ~125.0. Colossus carries nearly 170.0 Lorenzini pores across its widened snout, and cephalofoil, which scaled approximately ~168,800. Spatial and directional resolution porous array spacing of ~1 cm gives a dense 3D grid of sensors, can pinpoint field sources within centimeters, and map complex electric landscapes. Minimum detectable field threshold ~0.5 nanovolt/centimeter, enabling detection of muscle potentials and nerve currents in other animals. Practical scenarios: heartbeat signals ~0.1 pmv/cm at 1 miles, sensed out to ~2-3 miles. Buried crustacean twitches ~0.2 miles depth, detected through sand to ~0.2 miles depth, tracked to ~50 miles. Why so sensitive? Receptor density, ~170.0 pores percentage surface area. Neural amplification, hypertrophied electrosensory lobes boost sub-nanovolt signals. Modular integration combines passive Lorenzini-style sensing, DC/geomagnetic, with active knife-fish-style electrolocation, AC fields. With this blended electrosensory powerhouse, Colossus reads the ocean's hidden electrical whispers and magnetic guides, even in complete darkness. Colossus's electrosensory system can pinpoint micrometer-scale muscle currents and nanovolt-level electric fields over tens of meters, turning it into a living underwater radar and bioelectric microscope, far surpassing any natural predator. Scaling of an electric eel's shock if you blew its organs up to megalodon size 15 vm. Using the square cube law, all numbers are hypothetical. 1. Scale factor. Length scale equals 15m/2m/7.5x. Area scale equals 7.5/56 havertz. Volume scale equals 7.5 havertz/420V. End quote. Voltage increase, series stacks, lengthen with body length, voltage scales /860V. Quote, 7.5.6, 450V3 current capacity. Coronet eel peak current, PEG 1A. Muscle/membrane area scales, PA. Hybrid, shark current, PA 1A. In 56, PA 56A. 4. Instantaneous power and energy per pulse. Instantaneous power, P equals VXI 56 per 150 VX56A EX361 kW. Pulse duration ~2s. Energy PXP PX361 kW PX 0.002 SX720 J5. Practical implications. Stunning range. 6. 450V shock across dark water could disable large prey at a few meters. Defensive radius. High current and voltage generate a strong field, potentially disrupts remote-controlled submersibles. Behavioral control. Small, localized pulses, Δ10J, could be used as conditioning cues in containment. By scaling EWL electrocyte stacks with shark length, Colossus could fire ~6 kV at ~50A for ~700J per pulse. A built-in 361 kD stun gun, powerful enough to incapacitate large fish or marine mammals on contact.